Hello, everyone, and welcome to the first in what we hope will be a series of sessions where we touch on some of the interesting facets of cancer biology and how these pathways are being targeted by pharma companies for therapeutic development. My name is Sean Cullen. I'm Associate Director of BD at Insilico Medicine. I've got a background in science with a PhD in cell biology, and I've spent some time in pharma or indeed with a stint at Genentech here in the Bay Area where I'm still based. I joined the BD realm 3 years ago, and I'm now, as I said, Associate Director for BD within Insilico Medicine. I'm joined here by my colleague Keith Mikule. Keith, would you like to introduce yourself?
Sure, thanks, Sean. Yeah, I'm Keith Mikule. I'm Vice President of Business Development at Insilico. Got about 20 years of industry experience across small, large, and medium biopharma, from target validation through to approvals. I spent the last 5 years on business development side, trying to bring new medicines to the clinic. So pleasure to be here.
Great, thank you, Keith. So as I mentioned earlier, the goal of this session really is to introduce to you the concept of synthetic lethality and specifically, and how that relates to cancer biology and how this promising area is being targeted in the drug development space. So with that, I will bring up the deck that we're working on today. Okay, so synthetic lethality has been in the news a lot lately, Keith. Can you explain the rationale behind this concept? Because it is a little bit difficult to understand, especially for newcomers to the space.
Sure, it's all based upon redundant processes. And redundant processes are great for precision, but they're generally not very efficient. And so in the example for drug development, what we try to take advantage of is that normal cells must produce an exact genetic replicate to survive under stressful condition. So this requires redundant mechanisms for essential processes such as DNA repair and controlling gene expression. However, tumor cells evolve to divide rapidly, and to do so, they generally lose these redundant pathways so that they can grow quickly. And in some cases, this can create a therapeutic opportunity that we call synthetic lethality. Now, the diagram on the right depicts two redundant genes for an essential cell function. So normal cells can survive the loss of either gene. However, in tumor cells that have lost gene A on the right-hand side, using a drug to target gene B will selectively kill the tumor cells and spare normal cells.
Very interesting, very promising approach for potential drug development. But what do you think are the main advantages of this as a therapeutic strategy?
So there are two main advantages that I'd point out. And so, doctors always struggle to identify to match patients with drugs. And so in this case, this doctors can select patients likely to respond to a drug by testing their tumor cells for the lost gene. And so that allows for a use of a very selective and potent drug to target that gene without concern for effects on normal cells, which makes this approach much safer overall than other strategies.
Very interesting biology background there, Keith, and I think it's fair to say that this opens up a very promising space for drug development purposes. But what are the main examples of synthetic lethal interactions, and have they been targeted from a clinical perspective?
Yeah, sure. The poster child for synthetic lethality is the approval of PARP inhibitors. Um, these have been clinically validated to work best in patients whose tumors have germline or sporadic mutations to the BRCA1 or BRCA2 genes. Um, that which BRCA, importantly, functions in the DNA repair pathway called homologous recombination. And inactivating mutations in BRCA1 or 2 make these cells dependent upon a redundant DNA repair mechanism known as non-homologous end joining. So PARP activity is essential for non-homologous end joining. And PARP and molecules that target PARP1 or 2 are are needed to are needed for non-homologous end joining, and so that we by targeting BRCA mutant patients with PARP inhibitors, we can kill those tumor cells and spare the normal cells. Now, the prevalence of BRCA mutations are fortunately low, but it's a strong driver of tumorigenesis in breast, ovarian, and prostate cancer patients. And several PARP inhibitors have now been approved to target this genetic deficiency, which has led to great benefits for these patients worldwide, with sales that have reached $7 billion in 2023. And a couple of examples of drugs that have been approved in this space are olaparib back in 2014 and niraparib in 2017, and some others since then.
I think it's fair to say that PARP inhibitors have really been a tremendous success, both from the patient viewpoint and also from an R&D perspective, really opening up synthetic lethality as a biological pathway that can be really potently targeted by therapeutic molecules. Are there any other examples of synthetic lethal targets in oncology, and what kind of work is going on in this space?
Yes, there are several other approaches focused on the DNA damage repair pathway, but these strategies have smaller patient populations that are likely to benefit based on the prevalence of the biomarkers. A more recent and perhaps more exciting example is in tumors that have lost the MTAP gene. This occurs, unfortunately, in a much broader patient population. Here, the redundant pathway that we're targeting is MAT2A, or MTA-bound PRMT5, which has been shown to selectively shrink tumors in patients with MTAP deletion. Um, some of the drugs that are being developed in this space include entrants from IDEA and Amgen, as well as some earlier stage drugs from in silico. Um, Based on the prevalence of MTAP deletion, this may be the largest synthetic lethality opportunity identified so far. And so it is expected to have a large benefit across a broad number of patients.
Thanks, Keith. MTAP deletion does seem like a very promising therapeutic strategy, but why do tumors lose MTAP in the first place? And why does MAT2A or PRMT5 inhibition selectively target those tumors with MTAP alterations?
Yeah, so this gene is located on a chromosome region called 9p21, which is the most frequent deletion in all of oncology. And this region encodes two key tumor suppressor genes, so CDK inhibitors actually. So without these genes, your cells proliferate very rapidly. In addition to that, this chromosomal region encodes for interferon genes associated with inflammation. So the patients with tumors that have this deletion, the tumors tend to be very aggressive and growing really rapidly and are resistant to standard of care, including immune checkpoint therapies, resulting in very poor outcomes. On the positive side, there are standard laboratory techniques that could be used to detect this loss. So expensive diagnostic tests are not required. And there are actually two therapeutic strategies, as you mentioned, that are being tested in clinical trials. And that's shown on the right-hand side. So the goal for this strategy is to inhibit the PRMT5 gene, which is an essential gene and a key player in gene expression. This enzyme uses S-adenosylmethionine, SAM, to methylate genes and control expression. So that's the key function we're trying to inhibit. And then there's two ways we're approaching that. So we can target MAT2A selectively, and that reduces the SAM levels, which inhibits PRMT5. But also, the second strategy is to target PRMT5 that's bound to MTA. And in MTAP deleted tumors, very high levels of MTA accumulate and inhibit PRMT5. And by targeting MTA bound form of PRMT5, we can shut down that enzyme selectively in tumor cells, and kill them and spare the normal cells that do not have an accumulation of MTA.
Very interesting. Thanks, Keith. So a complicated pathway, but a lot of number of different approaches being used to try and drug this for therapeutic development. But I think it really does look to be set up very nicely from a drug development standpoint. In particular, you mentioned that MTAP mutations seem to be much more frequent than BRCA mutations. So what types of human tumors harbor MTAP mutations, and what kind of patient populations are we looking at?
The first clinical entrants in this space have developed their molecules in bladder cancer and in lung cancer. And what we've seen is diagnostic samples from bladder cancer patients and lung cancer patients have, in the case of bladder, about a 30% incidence of MTAP loss. But the patients that are being put on trials are those patients that have failed other therapies. And in that patient population, we're seeing almost 50% of bladder cancer patients, for example, have MTAP deletion. So again, this drives both aggressiveness and resistance to disease. And the same is true in lung cancer. So about 13% of lung cancer patients have MTAP deletion. But again, the patients that are actually MTAP deficient that are being put on trial, the incidence rate is higher, upwards of 25% of lung cancer patients. So again, just reiterating, it just really speaks to the concept that this MTAP deletion drives resistance to standard therapies, and on top of that, increases the incidence overall is increased, so it becomes a very large number of patients that could potentially benefit from targeting this pathway.
Yeah, I think it's fair to say it's a very promising pathway, especially in the context of, as you said, resistance to currently available therapies, especially in later lines. So very promising for patients. As I mentioned earlier, there have been some encouraging recent clinical successes in this space already. What are your thoughts on this emerging data, Keith, and how do you think it positions the field for future success?
Yeah, well, there's been some debate overall. Once these pathways were identified, there's been a very active debate on whether or not the MAT2A would be the most the better target, or whether PRMT5 could potentially be the winner. And what I would say is the data is very early. IDEA has a molecule called IDE 397. They're in phase 2. They've only evaluated about 27 patients that they've reported on so far. But what they've seen is a very high overall response rate, upwards of 33% of the patients that are MTAP deleted in breast cancer and breast and lung cancer. I'm sorry, bladder and lung cancer, have shown really robust responses so far, including a complete response. BR, the PRMT5, cooperative PRMT5 inhibitors from BMS and Amgen, these have been consistent in about a 20%, 21% partial response rate overall, and no complete responses have been observed so far, despite being tested in about 100 and over 160 patients. So far, while the data is early, as a monotherapy, MAT2A seems to have more efficacy than PRMT5 inhibition. On the bottom here, it's just worth mentioning that Pfizer's molecule is since discontinued. That one showed is a non-cooperative, non-cooperative PRMT5. So it targets both the tumor cells and normal cells. And in there, while there was some evidence of efficacy, the toxicity profile was very, very aggressive. So it was not an opportunity to develop beyond phase one.
Thanks, Keith. So yeah, I think it's fair to say some very promising emerging clinical data in both the PRMT5 and MAT2A space. Um, Silica Medicine actually has its own MAT2A inhibitor that was developed by our in-house generative AI-driven drug development platform, Chemistry 42. How is this program positioned relative to competitors, and what's the current development status?
Um, well, our MAT2A program is entering the clinic in 2025. And I believe we have an opportunity to both catch up to our competitors, but also to broaden that patient population. Um, as I highlighted previously, glioblastoma, for example, up to 50% is super high unmet medical need. And in those patients, upward 50% of GBM patients have lost the MTAP gene. And based on our non-clinical data, our molecule has an improved brain exposure, which may allow both MTAP deleted GBM patients to benefit, but also those patients in other indications that may have brain metastases. So there's a real opportunity here to provide a best-in-class approach to MAT2A inhibition.
Thanks, Keith. But Insilico actually also have a PRMT5 inhibitor, and that's at an earlier stage of development. What's the current development status here, and what's your take on the potential for combining this with MAT2A inhibition?
Yeah, the PRMT5 program is pretty exciting. It's about a year behind our MAT2A program. And what's interesting is that the preclinical data suggests that lower doses of each molecule, both the MAT2A and the PRMT5, allow for maximum efficacy. And so how we expect this will translate into patients is that we expect by using the combination, we'll have an improved safety profile in combination versus monotherapy. Because in monotherapy, so by using lower doses of both MAT2A and PRMT5, we can actually suppress PRMT5 effectively to sufficiently to trigger tumor cell death and spare those normal cells.
Thanks, Keith. So very promising developments in the synthetic lethality space, in the drug development space as a whole, but also at Insilico. Insilico actually has prioritized synthetic lethality as a therapeutic strategy, with a number of programs in development in addition to MAT2A and PRMT5. Could you touch on some of these other programs?
Yeah, no, I think the synthetic lethal approach is really exciting as, again, the benefit is killing tumor cells selectively while sparing normal. And anytime you can do that in a biomarker guided clinical study, it allows for a lot more benefit. And so we're looking to test a number of clinical hypotheses, including tumors those tumors that are chromosomally unstable with a program called KIF18A, as well as another DNA damage repair opportunity that allows for WRN inhibitors to demonstrate synthetic lethality in tumors that that have mismatch repair.
Very interesting. So I think it's fair to say some exciting programs coming down the tracks at Insilico. And for anyone interested in partnering discussions around these assets, could you outline Insilico's partnering strategy and how we see potential partners aiding developments of these programs?
Sure. Our goal is to build on our discovery expertise. We believe we have the most efficient discovery engine in biopharma. And currently we are averaging, for example, we are currently averaging about 13 months from target selection to preclinical candidate nomination. And so that we have the capacity to deliver about 12 preclinical programs per preclinical candidates per year. Um, all that being said, we're relatively small companies, so larger pharma companies and specialized biotech are better positioned to develop and commercialize our pipeline. Therefore, partnering with these experts allows us to efficiently bring value to patients. And so we're always interested in talking to partners that are interested in licensing our programs.
Thanks a lot, Keith. Very interesting biological pathway, both the synthetic lethality as a whole and specifically the MAT2A, the MAT2A PRMT5 space. As Keith mentioned, we're open for partnering discussions for the right partner. So please reach out to us at bd@insilico.com for partnering discussions. And with that, I'd like to thank Keith and all of you for joining. We both hope you have a great start to your 2025, and we look forward to seeing you on the next installment.